Greetings, everyone, and welcome to today's session for your Human Growth and Development online course. We're going to be covering the third week of development today, and just a quick heads up, this is going to be a relatively short session. It's just an introduction to the events that happen in the third week, but the extensive details of development and all the further changes that happen are actually going to be taken in the next PowerPoint presentation, which is going to be on the embryonic period. So this is just sort of like an introductory session into that phase of development. All right, so let's go ahead and get started. Okay, so just a quick recap. By the end of the second week, the developing embryo has the following features. It has established a maternal-fetal communication. If you guys remember, we had the appearance of the network in the syncytial trophoblast layer, and the maternal capillaries kind of opened into those spaces, and then the blood just fused into that network. It has two cell layers. Remember, we called it the bilaminar germ disc, and it was made of the epiblast and hypoblast. And surrounding these two cell layers, we had two cavities, which were the amnion or amniotic cavity above the epiblast, and we had the secondary yolk sac, which originated from the primitive yolk sac. Remember, we had the large extra cavity around this entire structure, which was called the chorionic cavity. And we have an extra cell layer, which was referred to as the extraembryonic mesoderm. Remember, you had a layer of extraembryonic mesoderm lining the germ disc and its cavities, and we had a layer of extraembryonic mesoderm lining the cytotrophoblast. And remember that part of the extraembryonic mesoderm did not develop a cavity within it, and that was found at the pole of the germinal disc. And we refer to that as the connecting stalk, which we said would end up becoming the umbilical cord. So, the main event or major change that happens during the third week of development is a process called gastrulation. Gastrulation is a process that happens under the influence of specific signals, and just to keep in mind that generally most of the changes that happen during development, whether it's the appearance of a new layer or whether something condenses to become something else, okay, this is usually influenced by signals coming from another place or another structure. In certain situations, we know where these signals are coming from. In other situations, this is something that is still under research, and people are kind of still trying to figure out where certain signals come from. So in this instance, the signals that we're referring to cause a new mass of cells to migrate between the epiblast and the hypoblast. So these two layers were originally stacked on top of each other, and then this new mass of cells just kind of pushes its way in between the two and separates them. And now, instead of having two layers, we convert the germ disc is converted into a three-layered structure. Now, since these three layers are now completely new things or new layers, we're going to have to give them separate names. So we can't refer to them as epiblast and hypoblast anymore. So once the three layers are formed, we refer to them as the ectoderm, which is the outer layer, the mesoderm, which is the layer in the middle, and the endoderm, which is the innermost layer. So those are your three layers formed during gastrulation. Okay, so with this picture, it's just basically showing you a cross-section of the germ disc. And usually the three layers, and when they're in picture form or video form, they're given specific colors just to differentiate them in terms of the changes that happen in each layer and what goes where. Okay, so normally the upper layer is in blue, the middle layer is usually in red, and then the lower layer or inner layer is usually in yellow. So in this instance, you can see the cells that were previously the epiblast. Okay, they're in blue, they're the blue ones, and remember that's just for description purposes. This is now referred to as the ectoderm. The inner layer, which is the yellow one that was previously the hypoblast, is now referred to as the endoderm. And then this new mass of cells, which is given a red color here, kind of pinches off and starts to migrate between the two layers, and this is referred to as the mesodermal or mesoderm layer. So the signals that initiate this change, or the signals that cause gastrulation to happen, come from a structure called the primitive streak. Okay, now streak is just basically a line, if we take the word in its basic meaning. A streak is a line. Okay, so that means that I have a line or a streak appearing somewhere. Okay, and then the appearance of that streak causes the gastrulation process to happen. The primitive streak is a longitudinal thickening that appears on the epiblast, usually around day 15 to day 16 of development. So it appears in between those 2 days. Okay, now it appears caudally, so it appears initially next to or near the tail of the developing embryo, and it grows cranially towards the head. Its cranial end has a circular expansion called the primitive node. There's also a depression in the node called the primitive pit, and it has a line on it called the primitive groove. Okay, so let's just look at that in picture form. Now, what we're looking at here is the embryonic disc from the top. Okay, so this is not a cross section, this is the entire embryonic disc from the top, and the only layer that we can see in this picture is the ectoderm, or the epiblast at this stage, obviously, because it still hasn't started the process of gastrulation. So this is the epiblast. Now, before we get into the formation of the primitive streak, I just want you guys to notice that one end of the disc is actually larger than the other. Okay, so you've got this upper end, which is kind of expanded and wider. This is usually referred to as the cranial end. When we talk about cranial, we're talking about things that are closer to the head, so higher up in the human body. So basically the head, the neck, the structures in the chest, basically the upper part of the body, okay, is closer to this side of the embryonic disc, all right? And then when it narrows down, and we talk about the caudal end, we're usually talking about things that are closer to the lower part of the body. So things like your lower limbs, your pelvis, your back, okay? This is the caudal end of the embryonic disc. So initially the primitive streak appears at the caudal end and it grows towards the cranial end. Okay? And in terms of structure, remember it has an expansion at its cranial end, so its end that is closer to the head has an expansion called the primitive node. There is a depression in that node called the primitive pit, and there's also a line on top of the primitive streak called the primitive groove. Okay, so remember that structure appears around day 15 to 16. It appears at the caudal end and starts growing towards the cranial end, and it is the structure that gives out the signals that causes gastrulation to happen. So the primitive streak is therefore responsible for the formation of the mesoderm. And eventually what happens to the primitive streak is that it degenerates and disappears by the 4th week of development. So please make sure you know the time span where the primitive streak is present. It appears on day 15 or 16, or just around that time frame, and by the 4th week of development, the end of it, it has disappeared and degenerated. All right? Now, once the 3 layers have been formed, okay, we have within the mesoderm, so remember that's the middle layer, a group of cells form like their own separate mass and arrange themselves longitudinally within the mesoderm. And longitudinally basically means along the length of the disc. Now, this structure, which is from the mesoderm, okay, is referred to as the notochord. And the notochord is important because, number one, it also gives out signals, and these signals are important for the formation of something called the neural plate. And we're going to see in the next PowerPoint presentation how this neural plate ends up becoming the nervous system. So basically, if there's a problem with the formation of the notochord, there's going to be something wrong with the formation of the neural plate, and therefore a problem with the development of the nervous system. And keep in mind that the neural plate develops within the ectoderm. So even though the structure that signals its formation is found in the mesoderm, the neural plate itself is actually found in the ectoderm. The other importance of the notochord is that with further development, it will end up becoming the vertebral column. All right, so those are the two important reasons that the notochord appears. Okay, so again, we're looking at a cross-section of the developing disc. What's the blue layer up here? Okay, good, that's the ectoderm. And then in the middle, the layer that's usually in pink or in red, that is the mesoderm. And then down here in yellow, what's this? Okay, good, this is the endoderm. All right, now if you look at the mesoderm, okay, you can clearly see that it's starting to divide into separate segments here. You've got this dark segment right next to the midline. You've got this more lateral or segment towards the end. Okay, we're going to get into the details of that next session. But I just want you to notice how there is a centrally arranged mass of cells right here within the mesoderm. Okay, that is the notochord. All right, and this is the structure that develops in sort of like a tube formation within the mesoderm, and it causes formation of the neural plate. Okay, so like I said, this is going to be a very short session. A summary of the events that happened during the third week is that you have the appearance of the primitive streak, which induces gastrulation, establishment of the 3 germ layers. You have formation of the notochord within the mesodermal layer, which therefore causes the formation of the neural plate. And let's just look at gastrulation in animation form. Hey, that rhymed. So you can just kind of get an idea of how it happens. Okay, so you're looking at the bilaminar disc. You've got the epiblast, the hypoblast. They've shown you where the different cavities are. Okay, you've got the cranial end and the caudal end, just like I explained. And this is the primitive streak. All right, it's appearing as a streak and depression within the epiblast, and now you're taking a cross-section of the embryonic disc. All right, and they're going to give you a close-up. All right, just real quickly. Remember, this is the epiblast and the hypoblast down here, and then, okay, you've got a new population of cells that just kind of showed up out of nowhere, and they're going to become the 3 different layers, okay? So first of all, you have the replacement of the hypoblast to give you the endoderm, and then you had the appearance of the new mass of cells in between the 3 layers and in between the 2 layers to give you now the mesoderm. Okay, and now it's just going to show you how the mesoderm kind of grows throughout the embryonic disc, and then you can see how the notochord forms in the middle of the mesoderm, and then the mesoderm kind of expands to fill out the whole disc. Okay, and you can see how with the appearance of the notochord, which was that green line in the middle, now we have a change that happens in the ectodermal layer, which is the appearance of this structure in the middle here, and that is the neural plate. Like I said, we're going to see this next session, but this is going to end up becoming the nervous system. Okay, so let's just look at the notochord real quick. Okay, so you can see here how there's the mesoderm in the middle. This is a cross section. You've got the ectoderm, the endoderm, and the mesoderm, and then the appearance of the notochord right in the middle of the mesoderm right there. Okay, and you can see how it's growing within the mesoderm itself. You can see how the cells are arranged centrally here to form the notochord. Okay, so those are the things that I wanted you to see just to kind of get an idea of how these changes happen. All right, so I think that wraps that up for this session. Okay, and as always, I'm going to leave you guys with a positive quote just to kind of get your morale up a little bit. And remember that if there is no struggle, there is no progress. I know that sometimes it feels like your life is not moving forward and you're stuck, and it doesn't matter how much you try, it doesn't feel like you're going anywhere. But all the struggle is worth it, and even though it doesn't look like it now, but there will be progress at some point. So just keep working hard, and hopefully everything will work out exactly the way it's supposed to. Thank you so much for joining me today. I hope this session, as short as it was useful and helpful for you, and I will see you guys next time.